morning, everyone. We are Group 2 and we will be presenting Exercise 2, 3 Thinking Concepts using MESQUIT. This exercise utilizes MESQUIT for the molecular analysis, sequence alignment, and generation of phylogenetic tree for the analysis of phylogenetic relationship of the umycetes. Umycetes, also known as water molds, are a class of eukaryotes that encompasses ecologically important animal and plant pathogens. They have filamentous morphology and ecological roles that are similar to fungi, and they were formerly considered to be a basal fungal lineage. Umycetes have come to be recognized as very distant relatives of real fungi, as morphological and molecular research have advanced since the latter half of the 20th century. Through convergent evolution and interkingdom horizontal gene transfer, they have simultaneously evolved comparable morphology and lifestyle. The study entitled Phylogenomic Reconstruction of the Umycete Phylogeny derived from 37 genomes was the first whole genome phylogenetic analysis for the umycetes as a class. This utilized protein data from 37 complete umycete genomes and 6 entire complete SAR genomes. The findings confirm Phytophytium's placement in the Peronospiralis, the downy Malleus placement in the Phytophthora genus, and the topology of clades within the Pythiales order. However, due to a lack of genomic data for several phylogenetic clades within Phytophthora, the resolution of the Peronospiralis as an order remains poor at some branches. This research established a solid foundation for future umycetes genome phylogenies based on more taxonomically broad datasets. In another study entitled Phytophytium, Molecular Phylogeny and Systematics presented molecular evidence that members of Pythium clade, as identified by Levesque and de Kock, belong to Phytophytium. The identity of Phytophytium as a separate phylogenetic entity is highly supported by maximum likelihood and Bayesian phylogenetic studies of nuclear ribosomal DNA and mitochondrial DNA cytochrome oxidase-ability 1, as well as statistical assessments of pairwise distances. Phytophytium is a morphological intermediate of Phytophthora and Pythium genera. The formal transfer of clade case species to Phytophytium genus is described, as well as a comparison with morphological similar species from the genera Pythium and Phytophthora. This exercise aims to establish a phylogenetic tree representing the species phylogeny, to analyze phylogenetic relationships from the generated phylogenetic tree through molecular data, to understand the complex evolutionary relationships, to utilize phylogenetic framework, and analyze biological phenomena.
The methodology will consist of three main parts, namely downloading of required software, blast analysis, and molecular analysis. Written below each part are the specifics. First, all required software must be downloaded, which are the text editor for your respective operating system, the Java software, Mesquite, and any working sequence alignment software on your device, specifically Muscle, PlusLW, or MAP. Next is the nucleotide blast analysis and blast N. This is done by inputting the given sequences on the enter query box and then clicking on the blast button. All first three identities of the sequences are collected. Next is a molecular analysis. For this section, first, we create a FASTA file, which consists of the in-group taxa and the chosen outgroup taxa, which in our case is the uricasmodic sunny from the study of Robidoux. Copy the FASTA text of the first accession code of each given sequence earlier, as well as the FASTA of your chosen outgroup, and then paste it on the text editor. After all FASTA texts have been collated, save it as a fasta.txt file. After this, a project file is created in Mesquite. Load the molecular data following the specified instructions and then save the project as a Nexus file. Taxa names can be simplified as well by double-clicking on them. Next is the sequence alignment. Click on Matrix and then select Align Multiple Sequences, check No Separate Threads, and then designate the file location for the chosen alignment software, and then click OK. After the alignment, check the resulting aligned sequences. In our case, the result did not produce a good aligned sequence data, and so we altered the sequence using the cleanup matrix. This feature reverse complements any sequence if necessary, and it also allows the multiple sequence alignment after any change in the sequence. Next is the construction of a phylogenetic tree. For this, we click on Analysis and choose Mesquite Heuristic Research. Other parameters needed are indicated on the diagram. After this, wait for the analysis, and once the generated phylogenetic tree is displayed, reroute the first tree by selecting the branch of the outgroup, and then store the first tree. After this is a consensus tree construction, following its specifics. Once again, as the consensus tree is displayed, reroute it as necessary. Lastly is a resultant discussion to be discussed by Mr. Africa and Mr. Isidro.
Hello, I am Miguel Alfonso Biafrica and I am here to discuss the BLAST-N results. Firstly, what is BLAST-N? It is a search engine that compares the user's inserted nucleotide sequences with its respective database so that it could match the user sequence with information. This is done by a heuristic approach, which means that it is fast and uses shortcuts. Our results is as follows. Our group's 27 sequences were searched in the BLAST-N database, and we have compiled the top 3 results for each sequence through this QR code, which can be seen in this QR code. Ultimately, from the 27 nucleotide sequences, 26 species were found. One of our bases is the expected value, which determines the probability of that result randomly appearing. The best E value would be 0. And the percent identity, which is the percentage of our inserted sequence matching with the database. Of course, there are limitations to the usage of BLAST-N. They are related to the limitations of the database. Specifically, if either the sequence that you are searching for is less studied, or when you put a sequence that is too short or too conservative that multiple species have that segment. 3 of these examples can be seen on the screen as Globosporangium macrosporum, Sylvaticum, and Irregulare, which is also matched with the genus Pythium. I want you to note down these 3 species when Francisco Isidro discusses the formulated phylogenetic tree. Because if we would use the genus Pythium, Globosporangium would no longer be part of the tree, and the clade Pythium would also become monophyletic. Because of this, it is important to review your BLAST-N results and to determine the species identity of your sequences.
Thank you, Migs. And now for the continuation of the results in discussion. Here we present the phylogenetic tree that we were able to formulate. Now we move on to the outgroup. So for our outgroup, the Urichisma dixoni, it is a biotrophic intracellular marine oocyte. It has the ability to infect multiple brown seaweed species. It can actually be seen here in the image provided above that it is currently infecting its brown algal host, which is the Ectocarpus siliculosus. So we were able to retrieve this marine oocyte and use it as our outgroup through the research conducted by Robidio in Carleton University. It can be seen here in the middle phylogram illustration that the researcher was able to utilize several oocyte species that are also present in our formulated phylogenetic tree. It can be identified that the halophytes, Saprolegnias, Phytopteras, and Phytiums are the species present in both the research paper and the formulated phylogenetic tree. So the most recent ancestor for the in-group and the node of divergence separating the outgroup from the in-group can also be observed. And additionally, the most recent organisms resulting from the divergence were also identified. These are the Phytophytium, Globosporangium, and the Phytium. So for the bootstrap values, 64% was formulated as the weakest score and 100% as the strongest coming from the value that includes the outgroup. So through rerouting, mostly of the bootstrap values are well and strongly supported.
And now for the conclusion and recommendations. Molecular sequences contribute to understanding the complex evolutionary relationships through the phylogenetic framework, which can be used to infer biological phenomena in future studies. The BLAST, nucleotide, and mesquite are specific tools to identify and analyze species as they search and compare similarities between nucleotide regions. However, it might still be necessary to double-check the results of the BLAST search. The species are under the phylum Umycota, and the identified outgroup is Urichisma dixoni, which may share some genetic similarities with higher-order Oomycetes but still genetically unique. Outgroups are important in the analysis of phylogenetic trees as they root the tree and produce an in-group of closely related species. Molecular data strongly supports the status of the outgroup, while weakly supporting the node at which the groups of most recent organisms diverged. Combined data analyses with the usage of concatenation methods to generate supermatrix generation is recommended. And finally, here are our references. And that's it for our presentation. Thank you.